Uh, I'm Dr. Osebe Clinton. I'll be taking you today through QSP-based dose selection for MABEL in first-in-human trials. This document is from or by FDA. It's a draft document to guide on the usage of quantitative systems pharmacology to estimate a starting dose that is safe but not subtherapeutic. What is QSP? QSP is quantitative systems pharmacology. What is MABEL? MABEL is the minimum anticipated biological effect level. So allow me to say that FDA posted these 2 weeks ago on their LinkedIn, and I found it very interesting, and I picked it up. I thought it would be it's something worth looking over. So I want us to go through these guidelines together, and yeah, if you're building in that space, if you are working for some of these companies, you're a CRO, you're a health data scientist, you're building these models. These are some of the things that you should be considering. Come along, come along. So, of course, the first dose is usually the hardest. I mean, it's not so obvious, but it should be obvious if you understand the whole drug discovery process. Something I've wrote on together with a whole history of QSP and some of the quantitative methods that are used in clinical pharmacology. You'll find the link attached below this video. So, when the dose is too high, of course, there's risk of grave harm to the patient, or rather to the participants or subjects who are exposed. And this is what definitely uh, pushed MABEL as a concept. Then, if the dose is too low, you'll have some therapeutic exposure. You have these participants who are taking such a high risk for almost zero or non-existent benefit. So then, what are some of the limits of the traditional methods that led to QSP being pushed forward or agitated for or being adopted? But before we look at that, allow me to help you in the definition of QSP. So in 2011, the National Institute of Health had a workshop and they released a white paper which brought a lot of attention to QSP methods. Okay. Sorry. So in the white paper, QSP was defined as an approach to translational medicine that combines computational and experimental methods. The two are combined together. I've wrote about this in one of our blogs. Go check out our blogs at Afia Data, and yeah, you'll get to read on some of the methods that or some of the benefits, or some of the amazing, amazing work that we've seen by combining computational and experimental methods together. So to elucidate, sorry, this is a method that combines computational and experimental methods to elucidate, validate, and apply new pharmacological concepts to the development and use of small molecule and biological drugs. That definition is so vast, but just know this, okay? That number one, this approach is based on mathematical models, where you have where you have models and disease characteristics and dynamic interactions between molecules and their receptors or target sites. This whole information is brought together and tried to be understood from the perspective, of course, of a biological system and used to determine our first in human trial dose. You do understand in the drug discovery process, there's a part where we have animals, mostly rodents, and then before we hop over to human clinical trials. So, historically, the first in human doses they were derived from animal toxicology, as we see from the slides, and these studies were based on no observed adverse effect level, severely toxic dose, where you have at least in 10% of the rodents, or HNSTD, highest non-severely toxic dose in non-rodent species. And then these doses will be converted to a human equivalent dose, and you'd get okay, a rough estimate, right? Now, these methods are very appropriate for many drugs, but they are limited to date, of course, for certain high risk products like immunomodulatory agents. Okay. Um, so let's look at in prose form at least, or in a lengthier version, how these methods fall short. So we have some drugs which are highly specific, they have rather highly specific targets that only exist in humans and not in rodents. So if you have the ability or you have the potential of missing these not ability, so you have the potential of missing these effects in humans because in rodents they might not elicit the pharmacodynamic response that you expect, but in humans you end up maybe having some unforeseen adverse effects that you never saw them coming. Okay, then some of these drugs also elicit steep pharmacodynamic responses, such as maybe T cell activation or cytokine release, and these can be difficult to predict in animal studies, like I've just said. Some relevant targets might be absent or differently expressed in animal species, what we sort of covers the point number one as well. So we have a limited translational relevance of animal toxicity. And also, finally, we have a standard allometric scale of doses not being able to account for such pharmacological differences because, anyway, I'll come to explain that point later on. So, what is MABEL? Like I said, this is the dose expected to produce a minimum biological effect in humans, and it's built from integrating all relevant data, starting from pharmacologic activity and receptor binding data from human cell lines, the pharmacokinetics of this drug, the whole ADME absorption, distribution, metabolism, and excretion, just to predict a dose just at the threshold where biological activity begins. So you do understand because these are quantitative systems, we must have some guidelines on how we apply them. Now, the FDA came up with this draft, and they sent it out so that the experts in the field and other practitioners can give them feedback. But let's look and see what our feedback might be by the end of this small video. So that's QSP defined. So we have looked at QSP versus traditional MABEL methods rather, where on the traditional methods you have in vitro pharmacology of the product, you have animal toxicology and allometric scaling. We are dependent on a single dose anchor and the effect and safety of the molecule or the drug are considered separately. Then with QSP, you have the integration of in vitro, in vivo, and in silico data. You have target engagement to downstream effect. It's kind of mechanistic. You have a simulation of a range of doses with uncertainty. I'll show you a good example in these slides where we can see a graph. If you have just go search on molecular docking and you'll see what I mean. It's really interesting. I love simulations because it's almost an if your parameters are correct, that is the caveat in this. If your parameters are correct, it's almost an instant cause and effect kind of thing that you can be able to witness and be hey. But so yeah, QSP another advantage is it balances therapeutic effect and safety together, unlike the traditional methods. So we have four general principles or guiding principles of QSP in first in human dose selection. Number one is the integration of all available data. Number two is that you have to account for species differences, like we've seen. Incorporate species-specific physiology, receptor densities, and pharmacodynamics to translate non-clinical data to humans. Then you should have mechanistic representation of biological mechanisms involved in drug response, the full sequence that is, and then finally understand your model uncertainties and do some iterative refinement. So let's look at the QSP modeling workflow. So at the very top, we have question of interest to be answered by the model. What exactly are we trying to answer? Yeah. So state clearly what the model will answer, the starting dose, the anticipated target dose range, then the planned dose escalation scheme. Is it every 2 hours? Is it and by what concentration? Stuff like that. Yeah. The context of use, of course, you should also capture that. Define the model specific role and scope, how the prediction answers the question, and what other information, uh, your decision, how you arrived at that first dose, what other information your decision is dependent on or it uses to determine that level of dose. Yes. Um, we also have the model risk assessment. Let's just look at it. We have the model influence and consequence. Under model influence, what are we talking about? The intended weight of the model outcomes in decision making. That is the weight given to the model in the decision versus other methods. You can see our table there, where you on the Y scale you have the rather on the vertical scale you have the high consequence, on the horizontal scale you have high influence. So the highest trigger is achieved at the top right corner, which has to be validated thoroughly because the consequence is high and the influence is high as well. That is the consequence of making a wrong decision. Okay. Um, and the low influence, no, nothing major. Um, High consequence, low influence, just moderate rigor, and as well as high influence, high consequence. So yeah, let's look at model. These are some of when we've talked about question of interest, we have talked about context of use, we've talked about model risk assessments to at least guide model eval and validation. Uh, then model construction, where the model structure represents the disease, biological mechanisms, and pathways related to the drug, right? So some of the critical components are target expression, the biomarker turnover, downstream pathways, uh, protein binding, metabolism of the drug, and localization. You have to test alternatives as well, evaluate alternative structures to see whether added elements substantially change predictions or not. So, model parameterization. Remember when I said something about simulations and parameters? Like you have to really get your parameters right. So obtain parameters from reliable sources. They ideally should be from direct experimental measurement. It should be at least backed by literature, so to say. For example, human specific targets, for example, for monoclonal antibodies and other human specific targets, characterize binding parameters using human derived cells or tissues. There's no way you're taking tissues, live tissues or cells from rodents and then, right? You get the point. Then for non-clinical data where you have animal data, you can estimate pharmacokinetics and pharmacodynamic parameters, but also you assess how well each mirrors human biology. I don't know about pigs, I don't know about rodents. Yeah, somewhere in there. So let's look at the verification, model verification, model calibration, and validation. You need to document both. That's the most important thing in these at this stage. Verification, equations and code free, and the code should be at least free of errors. Your equations, your simulation equations, if you are lost on what sort of equations we are talking about, um, they should they should check out logically speaking, right? There's no way if you have not for example, if you have not introduced any drug, we should not be seeing your simulation run like there's an effect and there's no drug at all. Okay, they should follow logic. High dose, of course, leads to target saturation. There's some scientific logic that it should follow. These are some of the things that then you use to verify if your model is making sense in the first place. Stochastic stability essentially means we should not deviate so much as these predictions are uh, stochastic in a way. We should not deviate so much from what we kind of expect. Okay, I'm not saying go and force your model to sort of make the sense that you want it to make, but it should be scientifically sound. That's all I'm trying to say. Then under calibration, document the methods that you have used to calibrate, rather adjust or adjust your parameters, and the resulting alignment with observed data. Like upon adjusting these two parameters or variables or three variables, we found this, and this was the maybe ligand molecule kind of behavior. Then validation should be driven by the context of use, sorry, and risk. Okay, compare against independent data. Same mechanism, clinical data adds assurance. What that means is look at scientific papers. Is there someone uh, or a group of people who have been, or is there work on the same? And do they match? Do they make sense? Right? Is your evidence sound enough with in comparison to already written work? So this is an example of a dose response curve. You simulate responses at multiple low doses around the anticipated start. Then the curve steepness, of course, will now tell us, okay, should we uh, increase the dose or start with a higher dose or maybe start with a lower dose than given. So if you have a shallow curve already, sorry, as we can see from the image on our left, a gradual response at low doses can support a slightly higher initial dose because you're okay. Maybe it will take too long for the effect to start kicking in, and you simply are trying to, or for the minimum biological effect to kick in, and you're simply trying to, balance between time of administration and the time that we start seeing the effects. Okay, you don't want to uh, you don't want to give your patient or rather administer your patient with a medicine or drug in this case, then maybe wait for what, 24 hours, 12 hours for the first effect to kick in, right? That's ineffective in a way. Then if it's steep, of course, start lower because a pronounced rise uh, will definitely mean serious escalation in a matter of seconds. You don't want that, right? Think about slow IV administration. Why we even do that in the first place? But now in this case, compared to dose and response, the first dose and the first noted response. So model uncertainty and sensitivity analysis determine how individual model parameters affect predictions. and dosing as well. Okay. So capture parameter sensitivity. Like I said, what variable did you change and what was observed? Then capture structural uncertainty by evaluating alternative biological hypotheses and model architectures. You might not be having the best architecture, or you might actually be having the best architecture. You need to capture that. Then quantify how your knowledge gaps or the knowledge gaps in your work or in your model affect confidence, and then test the robustness of the recommendation. These are simply limitations. Okay. Okay, so then we proceed to dose selection. Then we have early monitoring and model refinement. Of course, dose selection has this from that stage, it's a whole with the results on the table, there's a whole lot of um, it's a multidisciplinary decision. Yeah. So if there's a large divergence, for example, if QSP differs substantially from traditional methods, then demonstrate reliability via retrospective validation on similar mechanism compounds. Okay. Add safety margins to just because you anticipate severity, reversibility, and clinical manageability of the novel drug or the novel target. So when estimates are discordant and there is no clinical precedent, choose the most conservative starting dose. I think that's a no-brainer. Then in life-threatening disease, a minimal active dose above the absolute MABEL may be justified. So I think that's why there's some medicines which you find the doses are very different depending on the criticality of the patient, right? Uh, even I'll give a very simple example. Even in asthma, we don't manage asthma the same way, right? From the early onset to a point where maybe the patient has advanced uh, asthma. We do not even use the same agents, right? Let alone dosage. We keep on adding on these agents. So early monitoring and model refinement. A refinement essentially means you update the model on emerging data uh, to keep it up to date. Then part of the monitoring is comparing what you're getting, or rather comparing established studies, with your prediction. For example, you collect early pharmacokinetic and pharmacodynamic biomarker data and check whether the human response, okay, matches the model. What your model told you, or what results you got from that model. And now that you have already administered it to your first human subjects, or rather patients, you need to be collecting data as you go and then compare it to what your model told you. Does it make sense, or did your model fail? Is there some maybe adverse drug reactions that were not captured? Is there sometimes there is it's not only always about ADEs or ADRs. Sometimes it's about another therapeutic effect discovered that had been missed. Yeah. Then you of course submit a QSP study report and engage whichever agency. Now in this case, because it was the FDA, yeah, you engage with them as early as development allows. Yes, I think that will cover up for today. Allow me to end the session here, um, because after that we have already after that it's a whole other process now of regulatory process. After you're confident in your molecule, it has been approved internally, it has met all checks, uh, and it seems to be working the way you anticipated. Now, the rest is just the regulatory part, but the model is still uh, engaged, as you can see. These are some of the things that you need to be submitting during the sponsor meetings. But I think we'll cover this next time, and there's nothing much to say there because it also depends with your country's authority. Okay, so I'll be writing a blog on QSP so that I can capture the technical details that I could not capture in this video. Allow me to do that. And yes, it shall be attached on the link below this video. Everything QSP, the advances we have made so far, some of the examples used, and maybe just maybe talk about an example of these models and a process that maybe share a real life process that hmm, those will be 700 words and I don't know. We'll see. We'll see. Stay tuned.